So again, folks, we're continuing our discussion of viruses. We had started the introductory PowerPoint, and we had talked about some of the history of the discovery of viruses. We talked a little bit about their structure, and now we're focusing more on structure. So folks, you'll recall that we said the simplest virus is what was famously described as trouble wrapped in protein. So guys, so let's take a look at that trouble wrapped in protein. This is what we call a naked virus, right? And viruses have a protein coat called a what? A capsid, right? And a really important job of the capsid is it's going to protect what inside? Yeah, the viral genetic information, which is going to be what? Yeah, DNA or RNA. Good. DNA or RNA. Well done, you guys. And one more thing, you guys, about the capsid that we'll see. If I use this poor little virus is just literally falling apart here. Okay, so you guys, if we pretended that this was the protein coat right here, the capsid, what will be really important is for the virus to be able to invade a host cell, it has to have special proteins on its outermost surface, and these are falling apart, but these are adhesins, protein adhesins, okay? So for viruses to bind to their host cell and be able to infect it, they have to have on their outermost layer, they have to have special proteins. And in this particular virus, you guys, we're gonna show them almost like lollipops. These are going to be the viral adhesins. And folks, what do you think adhesins do? Like adhesive tape? Yeah, they stick, right? So we're going to see that the first step in viral infection, if this is the host cell, the first step in viral infection is that the virus has to attach to the host cell. And it's going to do that by using the adhesins that bind to complementary host cell surface receptors. So this little cup here is the host cell surface receptor. So a virus has to have adhesins that have a complementary size, shape, and charge distribution so they can attach to the host cell surface receptor. And that's the first step in infection, is attachment or absorption. And we'll come back to that. Okay. So in a naked virus, where will the adhesins be? On the capsid, right? And as a consequence, you guys, that protein capsid is relatively tough. So the bad news is with these so-called naked viruses, when they're shed into the environment, they can remain infectious for long periods of time. And that's bad news for us humans, right? Okay. Now we're going to contrast that, folks, to what we call envelope viruses. And an envelope virus, we're going to take our good old naked virus, right? So here's the capsid, here's the nucleic acid, and we're going to coat the virus in stolen host cell membrane. So this blue, folks, this is stolen host cell membrane. It could be cytoplasmic membrane, it could be nuclear membrane, right? For most of the envelope viruses we'll be discussing, usually it's going to be stolen host cell membrane, cytoplasmic membrane. So folks, and this is just goofy, me trying to make more toys. So you guys, if this bag represents the stolen host cell membrane, in an envelope virus, then, would it make sense that the adhesins would be on the capsid? Would that make sense? It doesn't make any sense, right? Because if the adhesins are on the capsid with the envelope covering them, the adhesins can't bind to the host cell surface receptors. So, folks, what would we find in the envelope of an envelope virus? The adhesins, right? And this means before the virus steals our cell membrane, it's going to modify the membrane by inserting what? Viral adhesins, right? And maybe some other proteins. So here's the viral adhesins in the envelope. Now, folks, remember, our model of cell membranes is a fluid mosaic model, an ocean of phospholipid bilayer, an ocean of phospholipids, the consistency of olive oil with membranes inserted, right? So we want to remember, you guys, those cell membranes, they're very delicate. right? So if I'm infected with an envelope virus and I shed it into the environment, anything that damages the envelope could be dry ink, sunlight, soap, right? Chemicals. Anything that damages the envelope means the adhesins can no longer function. So as a consequence, envelope viruses do not remain infectious in the environment for long periods of time unless they're protected by moist organic material. And that could be feces, it could be blood, it could be tissue, right? But usually once the envelope virus is dry or exposed to sunlight, usually the damage to the envelope means they will not remain infectious in the environment for long periods. Okay, good. So that's a really important concept, you guys. So what we have on the PowerPoint slide, folks, is now we can classify our viruses based on their genetic information and whether they're naked or enveloped. Okay, so let me just put a few of these up on the board. And then there's more slides, folks, where we actually show you models of naked and enveloped viruses and give you some further examples. But we'll just put this up here on the board right now. Okay, so if we have our viruses, we can classify them according to whether they are DNA viruses or RNA viruses. And folks, who will have the higher mutation rate? RNA viruses, why? Yeah, so the RNA polymerase that's going to make the viral RNA does not proofread, right? Good job, you guys. And now we're going to add, now we know a little bit more, we can further classify them as naked or as enveloped. Okay? All right. And I'm hoping that the ones that I put up on the board, I'm hoping we're going to have time to talk about them in more detail. Okay? So a naked DNA virus, you guys, is HPV. And that stands for human papillomavirus, commonly called the wart viruses, right? So there's one of the things I hope we'll have time to talk about is some of the HPVs, the human papillomaviruses, are what we call oncogenic. What does oncogenic mean? Oncogenic. An oncologist is somebody who studies and tries to help people with what? Cancer, right? So you guys, so oncogenic viruses are viruses that, if we get infected with them, increase our risk for which disease? Cancers, right. So hopefully we'll have time to come back and say there are specific strains of HPV that may increase our risk for certain types of cancers, right? So bad news for us, right? And furthermore, you guys, with the wart viruses, because they're naked viruses, like if I have a wart on my foot and maybe I'm at the swimming pool or at the gym, right? And I'm shedding these naked DNA viruses into the environment, are they going to remain infectious for long periods of time? Yeah, right, as a naked virus, right, they can, so bad news for us. A naked RNA virus, folks, is a poliovirus. Where does poliovirus replicate? In the intestine, so how will we shed it? In feces, right? So wherever the feces goes, the virus goes. So if the feces contaminates food or drinking water and we swallow it, what could happen? We could get infected, right? So it's fecal-oral transmission. And the fact, you guys, that poliovirus is naked virus, is that bad news for us humans? Yes, why? They can remain infectious in the environment for long periods of time. Good. All right. And then, folks, let's move down to the envelope viruses. So an envelope DNA virus it's a big family of viruses, folks, the Herpes virus families. And people are always, I think, like a little bit shocked to find out that chickenpox is a herpes virus, right? And we'll talk about, there was this incredible medical microbiologist. He had retired from the medical school at UC Davis, and he came to give a talk on sexually transmitted viruses. Very distinguished. And he started out the lecture by saying, now class, why are herpes viruses like diamonds? And I'm sitting there going, what? You know, I'm supposed to know this stuff and I don't. And he goes, diamonds are forever. So we're going to find out, you guys, that, those of us that have herpes, we have them for forever. We're going to talk about these lifelong latent infections. Good. And then another one, folks, we might barely have time to talk about is the hepatitis B virus. So these are viruses that like to invade which organ? The liver, right? And there's many different types of hepatitis viruses, you guys, so hopefully we'll have time to talk about the hepatitis B virus just briefly. And then, folks, the enveloped RNA viruses I hope we'll talk about are the influenza viruses and HIV. And again, folks, the only good news I know to share with you about HIV is, thank heavens, it's an envelope virus, right? So if it gets shed into the environment and if it dries up, it's not going to be infectious for very long, right? So that's really important that we share that with our patients and their families. You know, we educate the public. It's like HIV will not remain infectious very long, like on a countertop or a door handle. Okay, good. And folks, you can see on the PowerPoint slide that there's other envelope RNA viruses. I am almost sure we won't get to talk about Ebola, MERS, SARS, dengue, or chikungunya. But if we had another semester, I would love to talk about those as kind of emerging viral diseases. But we're going to run out of time. Okay, so let's keep going, you guys. And again, most of the follow-up slides are just kind of showing examples of these different types of viruses. So this is just a generic naked virus, folks. So the outer shell is made out of what? Proteins, good. And then where would the adhesins be, folks? On the capsid, right? So when naked viruses are shed into the environment, do they remain infectious for relatively long periods of time? Yes, they do, yeah. And then, folks, what is the purple in there? That's a genetic information, which is either DNA or RNA. Good job, you guys. Excellent. Can you name two naked viruses? HPV, right? Human papillomavirus and poliovirus. Good job. Okay. And just some examples, folks. In the upper right-hand corner, those are all infections caused by what, do you think? HPV, human papillomavirus. And folks, I want you to know that usually the HPV strains that cause like warts, this accumulation of cells, like on our hands or the bottom of our feet, those are usually not the oncogenic strains. Okay, and we'll hopefully come back and talk about those oncogenic strains. And also, folks, the good news is they now have vaccines against a lot of those oncogenic HPV strains, so that's good news. In the lower left-hand corner, folks, this is a photograph. This could have been maybe from the 40s or 50s before we had the polio vaccines. It's heartbreaking, folks, but in some people to get infected with the polio virus, it's not in all people, but in some people to get infected, the polio virus can invade the nervous system and cause paralysis. Paralysis so you can't walk, and even more importantly, paralysis of the muscles used in respiration. So a lot of polio victims had to live in these iron lungs, and the iron lungs, they go through cycles of positive pressure, negative pressure, positive pressure, negative pressure to help you passively breathe. But can you imagine? It would be so tough. Your life would be so tough there. I remember as a kid, my best friend in our neighborhood, I loved to go play with her because she couldn't walk. And so we would crawl everywhere. We would pretend we're puppies or kitty cats or horses. I thought it was great. She was like my best friend. But it was only later that I found out she had suffered from polio and she was permanently paralyzed. Yeah, so it was like, yeah, interesting, huh? Luckily, folks, we have polio vaccines. So this is the second infectious disease the World Health Organization is hoping we can eradicate so no child has to suffer from polio. And then, folks, the electron micrograph in the lower right-hand corner, this is a picture of a so-called Rhino virus, the cold virus, and we're not going to talk much about them. And the reason is usually the infections are not that serious. Usually they're just upper respiratory tract infections. But they are naked viruses, so why is it that when one person gets a cold, it can rapidly spread? They're naked viruses, right? So the viruses, when we sneeze or cough, they can remain infectious on inanimate objects. So if another person comes along and touches, contaminates their hands, and then rubs their eyes or their nose or their mouth, that's a good way to get infected. So envelope viruses, folks. And I like to ask on the exam, what is the source or the origins of the viral envelope? Good. Host cell membranes, right? It's coming from you or from the host cell, right? And then we want to remember, folks, that what must the virus do to that stolen host cell membrane before it's a functional envelope? What do they have to insert into it? Yeah, they have to insert their viral adhesins, right, at the very least. So they have to insert their viral proteins into the cell membrane that's going to be stolen, right? And some of those proteins will act as viral adhesins. On this one, folks, this is in this particular envelope virus here, this is the capsid inside with the nucleic acid. This is the stolen host cell membrane. You can see in this particular virus, they've inserted 3 different kinds of proteins in there. So the envelope proteins, we know for sure they have to function as adhesins, but we'll see there can be also additional functions by different envelope proteins. Okay. And then, folks, we do want to remember the good news about these envelope viruses, they don't remain infectious in the environment for long periods unless they're protected by moist organic matter. And the 2 examples here, folks, on the top picture, this is an example of, in the old days, they were called Herpes Simplex viruses. Now we're supposed to call them human herpes viruses, type 1 and type 2. And again, I'm hoping we'll have time to discuss them in more detail. But there are the human herpes virus type 1, if it read our microbiology textbooks, it would know it's supposed to cause infections above the belt, meaning mouth, face, eyes, fingers. And that human herpes virus type 2 is supposed to cause infections below the belt, meaning usually sexually transmitted infections, right? But you guys, we're going to emphasize this. Do viruses read our textbooks? Nope. So it's important that we share with our patients that the type 2 viruses can infect the mouth, the face, and type 1s can infect the genital tract, right? So remember, viruses could care less about our books, right? Okay. And then, folks, the electron micrograph on the lower right, this is HIV escaping from a host cell. And again, folks, the only good news I know about HIV is enveloped, so it will not remain infectious in the environment for long. Yeah, that's really important. Now, folks, I don't want you to panic. This is a table showing you the DNA virus families. You don't have to memorize them, but I want us to count them. Okay, so you guys, how many DNA virus families are there? 1, 2, 3, 4, 5, 6, 7. Okay, and folks, I inserted an N if the viruses in that particular family are naked viruses. Okay, and again, you guys don't have to memorize this table, but there's a point here to including it. So how many DNA virus families? 7, okay? And now, folks, let's take a look at the RNA virus families. How many? Well, I'll tell you, there's 15, right? And so you guys, there's more than twice as many RNA virus families. What would be a really intelligent explanation? Why is there such greater diversity amongst RNA viruses? That's right, the high mutation rates, right? So we see really genetically diverse RNA viruses. And that means they've been branching and evolving into these different families generation after generation. Okay. All right, folks, so I know most of you are mostly interested in human viruses, human viral infections. But historically, you guys, it was really hard to study human virus infection because in the old days, to study a human virus replication cycle, you would purposely have to inoculate humans or lab animals, right? And there's ethical, moral issues associated there. So our early understanding of viral replication came from the study of bacterial viruses, bacteriophage. So I know a lot of you say, oh, this is kind of boring, I really don't care about bacteriophage. But folks, you would be amazed at how similar replication of human and animal viruses, how many steps are really similar to how bacterial viruses replicate. So we can learn a lot from studying the bacteriophage, the bacteria viruses. And furthermore, folks, in understanding how bacteriophage replicate, it lets us understand why the use of bacteriophage therapy to kill multi-antibiotic resistant bacterial pathogens, this could finally be an answer to these pan-resistant bacterial pathogens, right? So this, I hope you guys read this article. I'll probably put some bonus questions on lecture exam 3 dealing with phage therapy, because I think this could really, really help save lives in the future. It already has helped save lives. Okay, so you guys, we're going to then talk about bacteriophage replication. And again, folks, this is, I'm hoping, I'm going to be putting this concept map up on the board, so I gave you my chicken scratches, hoping that might help. Take notes, okay? So folks, first of all, we're going to be looking at a group of bacteriophages called the Lytic phage, and an example is the family called the T bacteriophage. Lytic meaning when they infect a bacterium, they're always going to kill the bacterium by causing it to lyse, right? So this is kind of bad news for the bacteria. Oh, and furthermore, you guys, we have these cool models here. So let me grab my little models. So these are models of the Lytic replication cycle. And the square, you guys, who do you think the square is? That's a cell, that's a bacterium, and this is E. coli. So the bacteriophage replication cycle, these are all bacteriophages that infect E. coli, right? So there's our E. coli, and there's our little guy attaching, and then things are going to get really nasty here. Okay. All right. Okay. So to understand replication, of course, we need to understand the structure of the bacteriophage. So these T phage, they're really fascinating, guys. The structure is really complex. So they have what's called an icosahedral head. That's a capsid that's made out of 20 triangular faces made out of protein, and it's protecting the phage DNA. Most of the bacteria, I think the majority of the bacteriophage have DNA as genetic information. And then they have this amazing, cool helical tail, and this is going to act as a hypodermic syringe here. And then these amazing tail fibers. So, oh my gosh, you guys, it's crazy. Just think nature's doing this, right, out there. Okay, and take a look, folks, at this is called the base plate and these spikes. So the bacterium is going to use these spikes to anchor itself into the bacterial cell wall. The adhesives, folks, are the tips of these delicate little tail fibers. Okay. So what we'll do, folks, is first walk through the steps, and then I'll show you the cartoon. And hopefully you guys, I got most of the steps, kind of the little bullet steps here on your handout. So we'll go through them quickly, and then we'll look at the cartoon. So you guys, in any kind of viral infection, the first step is what? Attachment. The virus has to attach to those host cell surface receptors. And this helps us understand why some viruses only infect humans, or some viruses only infect cats, right? Because the virus has to have the adhesins that will bind to specific molecules on the surface of the cell, right? So a virus that only infects cats doesn't have adhesins that can bind to surface receptor on human cells. It also helps us understand what's called tissue tropism in viruses. Why is it that one virus that invades me will primarily infect my liver cells, right? And another virus that invades me will primarily invade my cells of the nervous system. And again, a lot of it just depends on what adhesins a virus has and what cell receptors are available. Okay, so we're going to attach. A fancy term for attachment, folks, I don't know why they just say attachment, but a fancy term for viruses attaching to a host cell is adsorption. Okay, and then, folks, in the next step, and this can be called entry or penetration, the phage is going to inject its DNA across the cell wall, across the cell membrane, into the bacterium. So the protein portion of the virus is going to remain outside the cell, but it literally injects the bacterium with the phage DNA. Once the phage DNA gets inside, it is kind of heartbreaking, folks, because when the phage DNA gets into the bacterium RNA polymerase transcribes the phage DNA into phage mRNA, and then the bacterial ribosomes translate the phage mRNA into phage proteins. So the bacterium is actively participating in its own death. You know, it's kind of like a Greek tragedy, it seems to me. Anyway, okay, so we're going to have synthesis, biosynthesis. Phage DNA is going to be made, copies of phage DNA. Phage proteins are going to be made. And then this is amazing, folks. So the phage proteins, they self-assemble into those capsids. And the capsids package little pieces of DNA from the cytoplasm. So most of the DNA in the cytoplasm of the bacterium at that point is going to be copies of phage DNA. But occasionally, guys, there's a little piece of the chromosomal DNA of the bacterium that will get packaged into a capsid by accident, and we'll talk about the results later. And then to make this crazier, you guys, the proteins of the tail are self-assembling. The tail attaches to the capsid, and then the tail fibers attach to the very bottom of the tail. I mean, it's like if you wrote this for a Hollywood science fiction movie, they'd say nobody would believe you. But it's happening out there in nature right now. And then the final step, now the phage have been replicated, they're assembled, they're inside the bacterium, they need to get out. So they release a lysozyme-like substance, right, which weakens the cell wall of the bacterium, and then you have, boom, lysis and release of the newly replicated phage, and then they just go and they bind to the neighboring bacterium and start it all over again. So, okay, so here's our E. coli. What would this stage be? So it's supposed to be like attachment or absorption. Good. And then, folks, what's the little phage going to do? Inject the DNA in there. Okay, and here they're showing the phage DNA is being copied, and they're not showing that the proteins are also being copied. I know this is hard to see, you guys. I apologize. But this is, see how the capsids have been assembled, right? And there's tail, so this is assembly. And then, I love this one. What's this? Lysis, the release, right? And all the phage are going to attach to neighboring bacteria. Yeah, Crystal.
How fast does it go?
Oh my gosh. I am so embarrassed to say, gosh, Crystal, that's really good. I just know, like, because in graduate school we worked with these little guys, and I know that overnight, if we infected E. coli in the afternoon, by the next day we'd have big plaques in the lawn. So, gosh, that's embarrassing. Let me look it up, right?
Does it vary between?
It does vary between the phage. That's right. Yeah, but I'm embarrassed, you guys. I know we'd have great big plaques, so that means the phage have gone through multiple cycles of replication in 24 hours. But instead of making something up, let me double check on it. Okay, give you the real. I'll put it on canvas. Okay, thanks. Yeah. So folks, again, this is just a cartoon of what's called lytic replication. So you guys, right now, what we're working on, and I apologize. So we're going to see there's two different types of replication cycles in bacteriophage. The simplest one, the one we just talked about, is called lytic replication. So we're working on this side of the handout, all right? So we've gone through the steps, and now we'll look at the cartoon. And I apologize, you guys. I'm going to walk away from the microphone here. So you guys, here's our E. coli, and the bacterial chromosome is in purple. Okay, and here's our little bacteriophage attaching. And the phage DNA, you guys, they put it in kind of a turquoise blue color. So we have attachment, and then we have entry, and we'll see a detailed cartoon, you guys, of how the phage injects the DNA into the bacterium. It's crazy. And then the phage DNA is transcribed and translated. There's enzymes that are going to start tearing apart the bacterial chromosome as a source of nucleotides to make copies of phage DNA, right? And then we have assembly. We have our capsids and tails being assembled. And again, folks, the capsids automatically package pieces of DNA. And most times it'll be phage DNA, but occasionally a little piece of bacterial DNA will get packaged into a phage. So now we have assembly is finished, we have release of the lysozyme-like substance, and now we have the release, the lysis of the bacterium. So you guys, in the lytic cycle, down at the bottom of your cartoon, okay, we go through the steps, and then it says results. So you guys, what's the result of the lytic replication cycle of phage? Newly replicated phage are released, right? And what happens to the bacterium? It dies, okay? And then, folks, the second thing there that I could ask you on the exam is who does this? I should say what, but I like to give them personality. So who does this? There's two groups of bacteria, folks. There's two groups of bacteriophage, right? And let me do a little of this up on the board. So phage replication. And I'm going to simplify it, folks. So we're just going to go lytic replication with the steps. Okay, it's a boom, 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 boom. We know that the bacterium is going to die. and the new phage are released. So those are two important results. Now, what we want to know is who can carry this out, which groups of bacteriophage, and there's two groups of bacteriophage we'll be discussing. The first group are called the Lytic phage, and our example folks are the T bacteriophage. They're called Lytic phage because they can only carry out lytic replication. They always kill their bacterial host. They always kill their bacterial host, right? And new phage are always going to be released. But folks, there's a second group of bacteriophage called the Temperate Faj. And let me just put it up here on the board, and then we're going to elaborate. We're going to describe them here in just a couple of minutes. So the Temperate Faj, and the classic example, you guys, is the Lambda Faj of E. coli. So remember, these are all E. coli phages, okay? All right. This is just details, you guys, of how the bacteriophage injects the bacterium with the DNA. So here, folks, just to get you oriented, this is the inside of our bacterium. This is the cytoplasmic membrane. This is E. coli, so it's gram-negative, right? Thin layer peptidoglycan. Here's the outer membrane. So our T phage is attached using the adhesins in the tail fibers. It settles down and anchors itself with the spikes on the base plate here. And then this is amazing, you guys. Then the tail contracts and it drives this hollow protein tube like a hypodermic needle across the cell wall and across the cell membrane. And in that contraction, it drives the phage DNA into the interior of the bacterium, right? It's like, this is crazy, crazy stuff. I'm just so amazed at that process. This is just showing assembly. So again, the proteins just know how to self-assemble into capsids. The capsids then will package a DNA. After the capsids have packaged DNA, then the tails will assemble to the capsid, the tail fibers will assemble, and then this is our mature bacteriophage ready to be released. Again, guys, it's just crazy. It just happens all by itself. It's amazing. Okay, folks, so now what we're going to do, this is where things are going to get a little bit more complicated. We're going to look at the second replication cycle of bacteriophage, and that's called the Lysogenic Cycle. And a significant difference, folks, between the Lytic cycle and the Lysogenic cycle is that in the Lysogenic cycle, the bacterium will not be killed. The bacterium is not going to be killed. Right, the Lysogenic cycle. And I think we're going to do this in reverse, you guys. Let's use the cartoon first, and then we'll show the text of the steps. And again, I've given you kind of a rough outline of the steps here. What you want to try to remember is, how does the lytic cycle differ from the lysogenic cycle? So I'll try to make a big deal about it. Here's the big difference here. Okay, so folks, in the lysogenic cycle, the first two steps are the same as in the lytic cycle. So this would be our little bacteriophage lambda, like little bacteriophage lambda. And lambda just has these little short little tail fibers. Folks, they don't have those nice long tail fibers. So lambda attaches, absorbs to the E. coli. What's step 2, folks? Entry. What does entry mean? The phage, injects the bacterium with the DNA. And this is where it goes totally different, folks. This is so wild. So in step 3, instead of the phage DNA being transcribed and translated and taking over the E. coli to the little phage factory, no. The phage DNA inserts itself into the bacterial chromosome. And it can stay there for generations after generations. The bacterium just figures the phage DNA is part of its chromosome. So whenever the bacterium is going to divide in two, it copies the chromosome, it copies the phage DNA. And so a copy of the chromosome and phage DNA are passed down to all the daughter cells. And folks, this is going to happen generation after generation after generation. This is crazy, right? So let's write this up on the board, folks. Okay, so we know that attachment is the same as lytic. We know the penetration, or the entry rather, entry or penetration is the same. The injection of the DNA is the same. But you guys, it's step 3 where things go really wild. So you guys, what happens in step 3? What happens in the lysogenic cycle that does not happen in the lytic cycle? Yeah, where's the Faj DNA heading? Yeah, good. The Faj DNA inserts into the bacterial chromosome. And you guys, now we've got new vocabulary. So when Faj DNA is carried in the bacterial chromosome, it's called a prophage. So we could say we have formation of a prophage. And I'll put a circle around it because it's important vocabulary. So okay, so we've described how the prophage is formed, but on the exam, I'm going to ask you going backwards. So you guys, what is a prophage? It's Faj DNA in a bacterial chromosome. Good job, folks. And the bacterium has a special name. Okay, so we're going to form a prophage, and now the bacterium has a special name. The bacterium is a lysogen, or another way of describing you guys, a lysogenized bacterium. So if I say you've got a lysogen or lysogenized bacterium, what do you know about it? What is it carrying? It's carrying a prophage. And what's a prophage? Faj DNA in the bacterial chromosome, right? Okay, this is just crazy. So, folks, in the lysogenic cycle, is the E. coli host killed? Nope. All right, so bacterium not killed. Are you going to get release of newly replicated Faj? No, right? There's no biosynthesis, no assembly, no release. So newly replicated Faj not released, right? And you're kind of like, well, what's the point, right? But you guys, is the Faj DNA being copied generation after generation after generation after generation? Yeah? Yeah? Okay. So this isn't a dead end for the bacteriophage. And indeed, you guys, let me go to the next slide. Sorry, I think this is just like the steps. Right. So again, folks, this is just emphasizing where the lysogenic cycle differs from the lytic cycle. The first 2 steps, attachment and entry, are the same as lytic, but it's the 3rd step. When we have formation of the prophage, does not happen in the lytic cycle. The bacterium becomes a lysogenized bacterium, does not happen in the lytic cycle, right? So this is the big difference here. And one thing we need to be aware of, you guys, is who carries this out? Who can carry out the lysogenic cycle? It's only one group of our bacteria. Who does this? So only the temperate bacteriophage, such as lambda, such as the lambda phage. It's only temperate phage that have the option to carry out either lysogenic or lytic. Because remember, you guys, the temperate phage, they can do lytic, but now we know they have this other option, lysogenic. So you're like, well, first of all, who cares, right? And secondly, well, what is going to decide which way the temperate phage are going to go, right? So the key to the lysogenic cycle, folks, is what's called a Faj repressor protein. And the Faj repressor protein, if you can imagine the lac operon, do you remember how the lac repressor protein blocked transcription of the beta-galactosidase and lactose transport gene? Do you remember that? How the lac repressor protein bound to the operator, and that blocked RNA polymerase from transcribing the lac operon genes? Yeah? Well, you guys, it's the same setup for the lytic cycle genes of the temperate phage. There's an operator, and if a Faj repressor protein binds to the operator, it blocks transcriptions of the genes required for the lytic cycle, right? And that throws the bacterium automatically into the lysogenic cycle, right? And again, you're like, well, who cares, truly? Well, the cool thing is, you guys, what happens if the E. coli gets hurt and the E. coli is starting to die? If you're that prophage, do you want to die with the E. coli, or do you want to get out of there as fast as you can? You want to get out of your E. coli, right, if it's dying. So that's what temperate phage can do. They can switch from the lysogenic cycle back to the lytic cycle in a process called induction. Okay? And the key events, you guys, in induction, let me go forward here. The key event in induction is that repressor protein, the phage repressor protein, is going to be destroyed. And that permits expression of the lytic cycle genes. That's what's letting the phage jump from lysogenic to lytic cycle. Okay? And a cartoon sometimes, you guys, is the best. All right, so let's go to the cartoon. So folks, let's follow this lambda phage. These are temperate phage. So they're going to attach, we're going to have the entry inject their DNA. And now they're going into the lysogenic cycle. The phage DNA inserts into the chromosome. We've got our prophage, we've got our lysogenized bacterium, they replicate, they pass on the prophage DNA, right? But folks, if there's some kind of damage to the E. coli host, and again, the classic example is DNA damage, right? It's almost like that's a signal that E. coli might die, right? And what will happen, you will activate an emergency repair system in the E. coli. And one of the enzymes that's activated kind of is, almost you could say is a mistake, destroys the phage repressor protein. And that permits the switch from the lysogenic cycle into which cycle? The lytic cycle, right? And what's the term for that switch, you guys? It's called induction. It's usually triggered by DNA damage. And the result is the phage repressor protein is destroyed. And that's what permits the phage to leave lysogenic and enter the lytic cycle. So you guys, here we see the phage DNA, it's actually cut itself out of the chromosome, and now it's triggered the biosynthesis, the transcription and translation, the assembly, and then the release. And again, you're kind of like, what the heck's going on here? So I always have to come up with a story. So my story is, folks, that the E. coli is a ship on the ocean, right? And the prophage is a rat on the ship. And the rat's so happy to be on the ship. It's warm, there's food, it's protected from bad weather, right? So the rat is very happy to stay on board the ship. But what happens, you guys, if the ship hits a coral reef or an iceberg? And now the ship is sinking. If you're that rat, do you want to go down with your ship? No, because you're going to die, right? So what do you need to do? You need to jump ship, right? Get out of there, right? Maybe, I don't know, climb onto a piece of lumber, jetsam and whatever. Okay, so you guys, that's the story I remember with the prophage. The prophage is like a rat on a ship. If the E. coli is damaged and is going to die, the phage DNA will be destroyed with it. So it needs to jump ship. And the only way it can jump ship is to switch into the lytic cycle. Okay? Now, again, you guys are probably like, well, that's not very interesting, really. But, you guys, it gets even more fascinating here. Because now that we know both about the lytic replication cycle and the lysogenic cycle, now, folks, we get to go back to horizontal gene transfer and look at two different types of transduction. Okay, do you vaguely remember transduction, you guys? Horizontal gene transfer in which the donor DNA is transferred to recipient bacterium by what? By who? By a bacteriophage. Okay, so you guys, what we're going to do then is at the bottom part of your handout where it says transduction, we're going to look at two different types of transduction, generalized transduction and specialized transduction. And I hope this will get a little bit more interesting for you here. Okay, so we'll keep our fingers crossed. So again, folks, if I asked you on the exam, if we're talking about horizontal gene transfer, what is transduction? In horizontal gene transfer, if it was like a short answer question, what would you tell me? So folks, remember in horizontal gene transfer, we always have a donor bacterium and a recipient bacterium. Good. And we know somehow the donor bacterium is going to reach the recipient, right? And there's different ways the donor DNA can reach the recipient. So in transduction, you guys, how does it happen? Good, awesome, you guys. Remember, in transduction, the donor bacterial DNA is transferred to the recipient by a bacteriophage, right? The bacteriophage is like the delivery truck, or nowadays we might say the Lyft or the Uber, right? It delivers the donor DNA to the recipient. Good, okay. But now, folks, now that we know about the two types of replication, now we can understand there's two different types of transduction. And again, folks, when I make up these concept maps, I'm just thinking up questions. You can think up your own questions to ask, you know. But what I came up with making this one was in transduction, may any donor genes, and remember it's a bacterium's DNA, be transferred to the recipient. Any random genes. May any donor genes be transferred to the recipient. And if the answer is yes, then this is called generalized transduction, right? And we're gonna back up, you guys. We'll back up and take a look at the cartoon. And in generalized transduction, folks, only the Lydic cycle is required. And because only the Lydic cycle is required, that means, you know, who can perform this? That means that both the Lytic Faj can perform generalized transduction, and who else can perform it? The Temperate Faj. Good job, folks. Okay. And again, you guys, we'll just back up to the Lytic replication cycle so we can understand how this is happening. It's just a mistake when the capsids are packaging DNA. Do you remember, you guys, how we said that if we're focused on a Lytic cycle here, folks, remember how we said that the capsids just automatically package little pieces of DNA, right? Usually most of the DNA of the bacterium by that time is going to be Faj DNA. But occasionally you guys, oops, I need a better. Sorry. Okay, this is kind of a nicer picture, you guys. Remember how we said in the Lytic cycle, the bacterial donor, in this case donor, chromosome gets chopped up? Well, occasionally there's a little piece of donor DNA floating around, and the donor DNA gets packaged into the capsids instead of the Faj DNA. And what's wild is that Faj, you might call it a defective Faj, carrying donor DNA, is still infectious. It can still bind to a neighboring bacterium, the recipient, and inject the recipient with the donor DNA. And then through homologous recombination, right, the donor DNA can get inserted into the recipient's chromosome. The reason it's called generalized, it could be any genes, any DNA fragment of the donor, right? Nothing specific. But in contrast, you guys, this is where it gets crazy. In the type of, here I had the slide here already. In the type of transduction called specialized transduction, only special genes are going to be transferred, right? So if we go back to the original question, you guys, may any donor genes be transferred to the recipient? And if the answer is no, then we're talking about specialized transduction, where only special, very specific genes of the donor will be transferred. And for this to occur, it requires both the lysogenic cycle and the lytic cycle. So which group of Faj, you guys, carries out both lysogenic and lytic? Only the temperate Faj, right? They're the only ones that can carry this out. And I know right now this is probably, again, I know a lot of this stuff, it's like, who cares, right? So let's use an example, folks, of specialized transduction in which a powerful toxin gene was transferred to a formerly avirulent non-pathogenic strain of E. coli. So now, you guys, we have all the knowledge to understand how E. coli O157H7 acquired that really harmful shigatoxin gene. Okay, so you guys, so I know this is we'll probably run out of room here. Are you okay, folks, if I erase some of this? It's mostly on the handout already. Okay, So you guys, we want to take a look at how specialized transduction, how this is a process by which bacteria can transfer toxin genes and antibiotic resistance genes to neighbors, okay? So let's take a look at specialized transduction. And now we have the details, folks, to see how exactly it was that this will be our E. coli. This is going to be our E. coli 0157H7 before it acquired the shigatoxin gene, right? Prior to World War II, this was a non-pathogenic bacterium. And this will be the chromosome. This will be our recipient's chromosome, our E. coli chromosome. Right? And now we need a donor. So you guys, who originally had the shigatoxin gene? It was a cousin of E. coli called Shigella dysenteria, the cause of bacillary dysentery. So this is going to be our donor. This is our donor. So let's do the donor chromosome. Okay, and folks, in my donor chromosome, this guy is the this was a bacterium that first evolved the shigatoxin gene. So let me just put a little piece of DNA here, you guys, for the shigatoxin gene, all right? So we want to remember, you guys, the shigatoxin, it shuts down ADS ribosomes. It inhibits ADS ribosome. What kind of ribosomes do we have? ADS, right? And that's going to cause all kinds of damage, right? And we'll talk about it in more detail here. Okay, so what we now know happened was that this Shigella dysenteria, it got lysogenized. What does that mean? Yeah, what's in the chromosome? Good, awesome, you guys. And you use the right vocabulary. Fantastic. Phage DNA was inserted into the chromosome. So what do we call the phage DNA in the chromosome? Prophage. Awesome, you guys. Excellent. Okay. So this Shigella dysentery was carrying a prophage, and this is important, you guys. The prophage had inserted right next to the shigatoxin gene. Okay. So what happened was we had induction, right? We had induction, and as a result of induction, remember, you guys, we say the prophage cuts itself out and then triggers a Lydic cycle. Sometimes the phage doesn't cut itself out precisely. Sometimes it cuts into the bacterial chromosomal DNA. So it ends up extracting itself, but it also takes a piece of the chromosomal DNA with it. So you guys, what do you think happened during this induction event? What got cut out? Yeah, the phage DNA and what else? Yeah, spot on, you guys. You nailed it. Right? So it was like a mistake, kind of a sloppy cut. And when the phage cut itself out, it cut into the chromosome and it carried then the shigatoxin gene. And then, folks, in synthesis, this becomes a template to make all the phage DNA. So all the copies of phage DNA included a copy of the shigatoxin gene. Does this make sense so far, folks? Okay, so then when we had assembly, all of the phage were carrying what? This recombinant DNA, right? So it's got phage DNA and what else? The shigatoxin gene. Awesome. I mean, it's not good, but you guys are just nailing it. And here's the shigatoxin gene. And then this phage, folks, could bind to who? These guys are close cousins, right? They're close cousins. So this phage, what could it do? Yeah, it can bind to E. coli and inject the E. coli with what? Yeah, the phage and the shigatoxin gene. And this is so wild, you guys. So what happened was the phage DNA inserts itself into the E. coli chromosome and takes who with it? The shigatoxin gene. Isn't that crazy? And the reason they know this is when they do the DNA sequence of the chromosome of E. coli 0157H7, they find the DNA sequence for the shigatoxin gene right beside what? The phage DNA sequence. So they know that it was the phage that brought the shigatoxin gene from Shigella dysentery to E. coli 0157. And again, you guys, and you should always be able, well, you guys, this is happening all the time. And that means that in your lifetime, in your professional lifetime, you are going to see the emergence of new toxin-carrying strains of bacteria, because this is not a one, this happens once and then it doesn't happen anymore. This is going on all the time. And that's why you have to have the prepared mind to know there's going to be new emergent toxin-producing strains of bacteria in my future that we don't even know exists right now, right? So again, folks, this is another reason for us to really respect the ability of bacteria to share genetic information, right? So we're worried not only about them sharing antibiotic resistance genes, but what else are we worried about? Them sharing toxin genes with one another, right? And again, this will contribute to the emergence of new bacterial pathogens, right? So again, you guys, being aware, being knowledgeable, that's a big part of the battle, I think. Okay, so folks, and I apologize, for me it was really hard to find good slides, cartooning, specialized transduction. I hope this makes sense, because in specialized transduction, what am I going to ask you about? How did E. coli 0157H7 acquire the shigatoxin gene, and what are you going to tell me? It was through specialized transduction. Good. And folks, I just want to do, and I apologize if this is repetition, but I feel so strongly about this that the public needs to be aware of the dangers of E. coli 0157H7. Okay? So, and again, folks, this might be repetition, and I apologize. So the E. coli 0157H7, where do E. coli grow, you guys? They grow in the intestines, right? And that means they're shed where? Or how are they shed? In feces, right? So what's transmission? Fecal oral, right? And an issue, folks, is that cattle can carry, cattle and other non-human animals can carry the E. coli 0157H7 in their intestines and not get sick, because their cells lack the receptor for the toxin. So we're going to find out that bacterial protein toxins, they too have to bind to specific receptors on cells. The cattle cells don't have the receptors for the toxin. So the E. coli can live in their intestine and there's no clinical signs or symptoms. But then, folks, when the cattle are taken to be slaughtered, right, and usually in the big slaughterhouses, the cattle know things are really wrong. So they go into fight or flight, they lose control of their bowels, and there's manure everywhere, feces everywhere, right? And the folks working in the slaughterhouses, folks, they're trying to be so careful, but when the hide is removed from the carcass, it's almost impossible not to get some fecal contamination on the carcass, right? So folks, and I know you're tired of me saying this, but just presume when you go to buy store-bought meat or poultry, you always presume on raw meat and raw poultry what? A fine frosting of fecal pathogens, right? Or fecal microbes, yeah? Right. So you guys, if you had if you eat beef, right, and you eat a big piece of beef like a steak, where are the fecal pathogens located? On the outside. They're not tissue invasive, you guys. It's just literally contamination. So your fecal bacteria are out here. So you guys, if you did a barbecue, this is a micro barbecue. If you sear the outside of the meat, will you kill the fecal bacteria? Yeah. So in theory, you could eat like a solid piece of meat, maybe medium rare, a little bit pink in the middle, right? But you guys, let's take the meat and let's grind it up and make a hamburger. And so now we've got our hamburger, and now where are the fecal bacteria? everywhere, right? So you guys, this is why it's so important that when you're eating ground meats, especially if you're feeding the ground meats to any high-risk group, so you guys, little kids, older people, anybody immunocompromised, never give them any kind of ground meat that's not what? Cooked. You want to make sure it's thoroughly cooked, right? Because now we've got fecal pathogens inside. So if you were to just cook it medium rare, right, what's a danger? The fecal bacteria inside are still alive, right? And we think that's what happened with the, you guys, I think you're too young to remember this, but there was an outbreak of E. coli 0157H7 and Jack in the Box. And so, and it ended up killing some kids. That's the problem, you guys. This thing is deadly. So they think either the hamburger wasn't cooked correctly or there was cross-contamination. Maybe, and maybe you guys, maybe you've seen this where this is the plate for your raw hamburgers, right? And maybe you're barbecuing and this is your spatula, right? So you cook your hamburgers and then you take this and put them back on that plate. It might be cross-contamination after they were cooked. But you guys, with E. coli 0157H7, right, if the kids are really vulnerable if they get infected. The E. coli's in the intestine, right? We have the Shiga toxin. The Shiga toxin is absorbed into the blood vessels, causes blood vessel damage. So what's an early sign you hope you might see? A bloody diarrhea. And again, folks, I would consider that a medical emergency. I'd want to get my kid to the doctor as soon as possible, right? And in some kids, it gets worse because what can happen from the blood vessel damage, what can happen to red blood cells? Yeah, you can get, your erythrocytes can lyse, right? And that can lead to a hemolytic anemia. There's not enough red blood cells to deliver oxygen to your tissues. What happens when your cells don't get enough oxygen? They start shutting down, right? And then the toxin can circulate to the kidneys and cause kidney damage. So you can get kidney failure. And one sign of that, you guys, is increased blood urea, because it's not getting filtered properly. So when we have multiple problems going on, we call it a syndrome, right? So you guys, what kind of syndrome can E. coli 0157H7 cause? HUS, and what's the H stand for? Hemolytic uremic syndrome. Good. Can this kill kids? It sure can. So again, folks, it was originally known as hamburger disease because it was associated with ground beef. But in the years since, now we're starting to see it in, they say leafy green vegetables you might use in salad. So any vegetable, you guys, that's not cooked, any fruit that's not cooked, if there's fecal contamination, right, and E. coli is present, that can be a source, right? So you guys, this guy, it's in California. And just keep your eyes and ears open because there'll frequently be recalls, right? There'll be recalls because they've identified that some food has been contaminated. One of the big outbreaks that I can remember here in California, it was several years ago, but it was in the bagged spinach. And what they finally discovered was wild pigs who were colonized with the E. coli 0157H7 had broken into the spinach fields, were having a nice, lovely spinach salad and pooping as they went. And so their feces had the E. coli 0157. The spinach was harvested, it was washed, but sometimes a wash water, you guys, if you add feces, then you're washing the spinach in fecal contaminated water, right? So those bags of washed greens. Yeah. And then they put them in a nice bag, which is like a nice incubator. Anyway, you guys, so just keep your eyes and ears peeled for good old E. coli 0157H7. Cooking will destroy it, you guys. So like if you have contaminated spinach, but you cook it, the E. coli is quickly killed by cooking. It's just something that's eaten raw or contaminated after it's cooked that we worry about. Okay, all right, you guys. So, and again, folks, I just wanted to stress that the comparing and contrasting the bacteriophage replication cycles, those are great exam questions. comparing and contrasting generalized transduction to specialized transduction, wonderful exam questions. Comparing Lytic Faj to Temperate Faj. Those are all wonderful exam type questions. And so folks on Canvas, I think I put a table comparing and contrasting Lytic replication to lysogenic replication. So I think that's on Canvas. Some people said that was helpful in preparing for these compare and contrast type questions on the exam. Okay, all right, you guys. So what we're doing then is we're going to use our understanding of bacteriophage replication to explore how human animal viruses replicate. And folks, just before I move on, we're kind of running out of time, but folks, if you can read the Scientific American article on the renewal of interest in bacteriophage therapy to kill multiple antibiotic-resistant bacterial pathogens, I'll try to have some bonus questions on exam 3, because I think this is really good news for us, right? And furthermore, folks, maybe you recall at the end of microbial genetics, we passed out some 3 bonus extra credit topics that would also be on lecture exam 3, and that was CRISPR. And folks, CRISPR, it's amazing. It's a type of specific acquired immunity bacteria have evolved to protect themselves against bacteriophage. And now it's famous because it's being used to do like really precise gene editing, right? But it originated, you guys, it's amazing. It's a type of specific acquired immunity bacteria have evolved to protect themselves against who? Bacteriophage, right? It's just mind-blowing. So those will be some additional bonus questions on lecture exam 3. So you guys, should we just wait until Thursday? Yeah, I know, you guys look so tired. Okay, so you guys, half of you have a good rest of the day, another half will see you at 1, okay? You guys take good care. Oh, and folks, if you want, I have, and you don't need to grab them today, but I have handouts for like the next, like viruses part B, I guess you'd call it. I'll absolutely bring them, you guys, on Thursday, but if you want to grab them early, please do. And I'll move the models here so you can reach these a little bit easier and better.